Welcome to the final podcast for day 18, chapter 11. In this podcast, we're only going to talk about objective 4. It involves apoptosis. Apoptosis is a very complex field, and we could spend a whole semester talking about it. But we just want to introduce you to this concept for right now. So this will be a fairly short podcast. Okay, so let's go ahead and get started. All right, remember I said we're going to talk about apoptosis. So let's go ahead and write that here and give it a quick definition. Apoptosis is a pathway that leads to programmed cell death. This is very important. We see programmed cell death that helps a lot of processes. For instance, it's what allows the webbing between our fingers to go away during our embryonic and fetal development. It's what allows our sunburned skin to peel away. These are all examples of programmed cell death. So it's very important. And this is much different than just cell death where the cell just kind of lyses and falls apart. This is very coordinated and the response is important. So on the previous podcast, we talked about signal transduction, these signaling pathways. And that's really what this is. So let's go ahead and draw another cell real quick. So here's our cell here. I'm going to draw this one a little smaller than on the last podcast. And we still have to have that first step of reception. In this case, let's draw this receptor like so. And we're going to call this a death receptor because it's going to signal death. It's a pretty ominous name, but that's what it's called. And then what binds to it is called a death ligand. Kind of scary terms here today. Death ligand. Or we could call it a death signal. So the death ligand or death signal comes in here and binds to the receptor. When it does this, it gives us some pathway, the transduction of this signal inside the cell. We don't need to get into the specifics for this class here, but in this pathway, it will take this protein here, and we don't have to give it a name, and it will inactivate it. So the signal comes in here, hits this protein, and it inactivates it. And the process of inactivating this protein, we can think of this protein that just got inactivated over there in blue. I'm drawing a mitochondria here. This protein has been keeping the cell alive, but now we've inactivated. And there are other proteins involved, seen in this cell, that have been just sort of waiting for this cell to become inactivated, the one that's keeping it alive. And these ones are going to signal it for death. Okay, there's several of them here. And like I said, they've just been waiting for this to happen. Now this pressure is relieved, and now they can go on and initiate programmed cell death. Some of the things that they will do, and let's just list them over here, is they will cause, let's list the first one here, will cause organelles like the mitochondria. They will also cause the DNA inside the nucleus to fragment. And they do some other things, but let's just close with this last one. They will cause the entire cell to fragment. Or I'm going to use the word breakup because that seems to be a little more appropriate. And what I mean by that is this large cell here will now appear as small fragmented parts of the cell. Within these parts of the cell that have just been broken up, you may find parts of the cell membranes in here. You may find parts of the mitochondria that have been degraded, as well as the DNA. But the cell is, at this point, for our practical purposes, dead. But now we have to get rid of it. And the way that these cells, this broken up cell is disposed of is by using another large cell, quite large cell, in our body called a macrophage. It's part of our immune system. And I'm just going to draw it as kind of a really large cell. And what it will do is it will come along here and engulf all of this up. And it'll bring all the little pieces of the broken up cell that just went through apoptosis into the macrophage. So I'm just going to draw a couple of these in here. And once they're in here, they're going to go into the lysosome and be degraded. This is a much cleaner way to kill a cell than just to have it lyse open. And when I say lyse open, like the plasma membrane gets holes in it. The reason this is better is, well, in this case is better, is that when you just poke holes in the plasma membrane, you can cause an inflammation response in the body, and then there's a lot of problems associated with inflammation. This prevents it all that inflammation from happening. This is I'm going to list a few examples of apoptosis that is very beneficial to us. This is how we get rid of cells that are cancerous. So cancer cells are destroyed. Another one is during development. So I'm just going to write development. But there are many examples in development where apoptosis is important. The simple one I talked about with removing the webbing from in between our fingers and our toes and removing the tail that we have during embryonic and fetal development. These are important things during development, so we're born the way we are. There are other examples in development as well. This is also important during infection, similar to cancer. So during infection, what will happen is sometimes viruses or bacteria will enter one of our cells, and to get rid of the bacteria or virus, the cell undergoes apoptosis, and that way killing itself, but also getting rid of the pathogen. So that's important. And the last one I'm going to say is for protection against DNA damage. So if a cell has too much DNA damage, it will be signaled to die through apoptosis. If you remember last week when we talked about the cell cycle and how it's regulated, we said before a cell enters S phase, it checks for mutations, and before a cell enters M phase, it checks for mutations. But we said we didn't really say what happened at that point. But if there's too much DNA damage, instead of going through S phase or continuing to M phase, the cell will enter apoptosis. And this is exactly happens when your skin peels when you have a sunburn. The UV light from the sun causes immense damage to your DNA. And often what happens is the cell says, there's no way we can fix this much DNA, so it just kills the cell. It's much better to do that than to allow a cell to continue to grow and accumulate mutations. All right, well, that's it for this podcast on apoptosis, and it ends chapter 11 material. If you have any questions over any of these podcasts or any of the material in this chapter, please make sure you come see one of us. Until then, we'll see you in class. Bye.